Good everyone and doctors. Today we're gonna present Mindmap of isolation, culture and functional characterization of Glia and endothelial cell from adult pig brain. So before we presenting our Mindmap, we would like to introduce ourselves.
Hi everyone, my name is BAKHTIAR BAHARUDIN and my metric number is 205782.
Hi everyone, my name is NURUL SABRINA. My matrix number is 207679.
Hello everyone, my name is PONNHMALAR and my matrix number is 205703.
Hello everyone, my name is SITI BINTI MOHAMADY and my matrix number is 207942.
Hello everyone, my name is GUAN. My matrix number is 206974. So we will start from the introduction part. DIYANAH, please. Okay.
Thank you, Egan. Can you zoom a little bit for me? All right, thank you. Okay, for the introduction part, we're studying about the glial and endothelial cells from adult pig brain. This primary culture of glial and endothelial cells is crucial for basic and translational neuroscience research. And this is because it better mimics the human central nervous systems, human CNS, and also the dysfunction of glia and endothelial cells will cause many human CNS diseases. And from the article, the adult pig brain tissue are easily accessible because it doesn't need to kill the animal and it is closest to the human brain. And pig is also the closest to human because it shares similarities of organ functioning, anatomical localization, and also gene pool sharing. However, few substantial differences occur between human and rodent glia in terms of its morphological and functional terms. In this research, primary cultures of oligodendrocytes, microglia, astrocytes, and also endothelial were generated from the same pig brain tissue and grown for 8 weeks. Okay, for glial cells, what is glial cells? It is essential to maintain the hemostasis, to protect the neuron, and form myelin in the central nervous system. And glial cells make up of 50%, more than 50% of CNL cells. So in the CNL cells, we can actually see the microglia, which is the brain resident macrophages and mononucleophagocytes that is crucial for CNS immune response. Next, we can found oligodendrocytes in CNS. It is myelinated and it offers supports for the neurons. Next, we can found astrocytes, which is vital for nutrition of neurons. And it is crucial for hemostasis in the extracellular space, as well as it important for repair mechanism. Next, we can also, instead of the glial cells, we can found another cells, which is a type of connecting cells called endothelial cells and pericytes. These cells are important for the formation and maintenance of the blood-brain barrier. So I pass to the next presenter, please. Thank you.
All right, thank you for the materials. Since it is impractical to put all the materials in the mind map, so I will speak briefly for the smaller parts until the bigger parts for the materials needed in this experiment. So the first part is isolation and expression of specific cell types. So under this part, there are another 3 smaller parts, which is dissociation of pig brain to make cell suspension. The materials needed are PBS, FCS, HBSS, and also the important one is the pig brain. The second small part is myelin removal from cell suspension. Isotonic Perco, DMEM, one-time BDM are needed. It's for the separation of glia cells. The co-separation buffer, FCR and blocker, magnetically labeled CD11B, and also CD31 plus are the materials needed for under the separation of glia cells. Next part is for the cell culture. There are 5 small parts under the cell culture. The first one is preparation of plates. So the materials needed are 100% of HCl, 70% of ethanol, polyl-ornithine, laminin, and also the distilled water. Next is culturing cells. For the culturing cells, we need to prepare the glial and endothelial cells, and also one-time BDM, FCS, as well as ARAC. Next is flow cytometric analysis. The materials needed are FACS buffer, one-time BD cytoperm solution, one-time BD perm buffer. For the real-time PCR, the materials needed are only simple, which we only require RNA as well as Taqman probe. Next one is immunocytochemistry. The materials needed are one-time PBS, 3.7% formaldehyde, 0.2% return X100, FcRn blocker, one-time PBST, and also 10% goat serum. For the last part is TNF alpha ELISA. Also under TNF alpha ELISA, there are another 5 smaller parts. For the first one is in vitro myelination assay. In this part, neuronal culture as well as oligodendrocyte culture are needed. MAP2, MOG, CD11, which is the specific antibodies, are needed. The next one is in vitro tube formation assay. Calcine AM green, calcine red, and H342, which is the dye, is needed in this part. One-time BDM, 1% FCS, and also 65 micrometer of suramin. Second one is in vitro neuronal survival assay. So ornithine and laminin in astrocyte media need to be prepared, as well as HBeGF. So the third one is cytotoxicity assay with AQP4 positive NMO serum. In this part, only 2 materials needed, poly-L-ornithine and laminin coated cover slips, as well as 10% of the human serum. For the last one is microscopic analysis, which the materials needed are inverted fluorescent microscope and also ISO vision software. That is all for the materials.
All right, I'm gonna continue with methods. Since Sabrina has already gone through the methods, I'm just briefly go through it again. Right, as mentioned by Sabrina, there are 3 parts of this practical. Can you zoom in a little bit? Yeah, the first one is the isolation and separation of specific cell types. There are 3 steps beneath these steps, which is dissociation of pig brain to make cell suspensions. Pig brain from 2 to 3 months old animal was obtained from the slaughterhouse. They use pig brain from animals which were slaughtered for meat production, so no animal was killed for the study. Thus, no approval by Animal Protection Authority or ethic board was necessary. It is followed with the myelin removal from cell suspensions and also separations of glia cells. Different cell type was separated by lineage specific labeling with antibodies and magnetic field based separations. And next is cell culture. All the separated cell fractions were cultured in a similar condition, but in the different cell type specific media. The surface of the cover slip plates or flasks were coated with L-ornithine and laminin. In these cell culture steps, there are 1, 2, 3, 5. There are 5 steps beneath it. First one is the preparations of the plates or flasks, followed with a culturing cell. flow cytometric analysis, real-time polymerase chain reactions, and immunocytochemistry. And for the next part is, can you zoom it next? Yes, TNF alpha ELISA. ELISA was carried out according to the manufacturer's protocol, porcine TNF alpha duo set ELISA from R&D system CAT. Cell culture spermidine were measured in duplicates at a dilutions of 1 to 10. In this part, there are 4 steps, which is first in vitro myelination assay, followed with in vitro tube formation assay, neuronal survival assay, cytotoxicity assay with AQP4 positive NMO serum, and lastly, microscopic analysis. After mounting the cover slip of the slides with prolonged diamond antiferrite mounted with the API, the slides were dry and scanned under inverted fluorescent microscope. Cell observed HS from ZIS at 20 times, 40 times, or 63 times magnifications. Images were captured at Exio Vision software. For each staining of different cell types, 600 to 1,000 cells were counted. Blinding of the scientists who performed the counting was performed for at least one of the replicate yielding for the same result. So I think that's it for the methods. I'm going to pass to the results. Thank you.
Okay, thank you, Bhatia. Okay, let me explain the finding of the this article. Okay, the flow cytometric analysis with the lineage-specific antibodies showed the purity of isolated cell fraction. For example, the CD11B expressed CD11B but not MOG, and CD31OC-FAP and the O4 sorted fraction are expressed. The cells were cultured in specific cell culture media where this medium helps in their growth and differentiation of the cells. The cells were observed by immunohistochemistry for the expression profile and to find out their purity. The triple negative fractions express GFAP based on the result. The results are confirmed by reverse transcription PCR, RT-PCR with the lineage-specific probes. To determine the morphological changes and expression profile, the cultures were cultured for 8 weeks, and these were the oligodendrocytes. After 2 weeks of culturing in a specific cell culture medium, 95% of the cells from the CD11B expressed CD11B and IBA1 but give negative result for other cells. And based on the outcome, 95% of the O4 sorted cell expressed the oligodendrocyte marker, while 80% of the CD31 expressed CD31 cells. 60% from the negative selected fraction expressed erythrocyte markers. The result can be divided into morphological and expression profile also. Oligodendrocytes and erythrocytes show increasing cell processes and branching, which shows the cellular differentiation. Mangrolia and endothelial cell did not show any changes. Finally, the expression of MBP for oligodendrocytes, GFAP for erythrocytes, and CD68 for mangrolia, and PCCAM3, and also known as CD31 for endothelial cell, remain stable even after the 8 weeks of culture. These are the results of the article. Thank you. Now I'll pass to GUAN for the discussion.
All right, thank you, Mala. So basically, the discussion divided into 4 parts. So first of all, the discussion mentioned about the primary gland and the endothelial cell culture has been beneficial to study the function and this function of this cell in vitro. And it also mentioned that these 2 tissue has their specificity. But however, for the rodent brain tissue, it's not ideally because due to its morphological, transcriptomic, and the proteomic differences. Well, for the pig brain tissue, it was ideally because, first of all, it shares more than 90% of genetic elements with human. And then in terms of macroscopic structure like size and weight is also share the similarity. And while for the microscopic structure, the gray or white matter ratio and the glial complexity are similar with the human cell. And beside that, it was the pig brain tissue was ideally because it allows isolating a large number of cells from a single brain, because the large size of pig brain and the easy availability of the tissue. And then the second part about the discussion mentioned is about another technology, which is the generation and maintenance of human induced pluripotent stem cell, iPSCs. So what is this technology? It's actually an alternative strategy to study the mechanism and pathophysiological of many diseases associated with gland cell or endothelial cell. But however, the journal is mentioned that this method is very expensive. It is a very expensive method, and it's very time consuming, and it's reprogramming of cells to iPSC is not trivial and require high expertise. And then it's also the differentiating iPS cell into 3 or 4 different mature cell types simultaneous is not an easy process. So this journal state that the method provided in the journal is more easier than this technology. So the third part of the discussion is about it compared to other study in protocol for purification and culture of mature oligodendrocyte from adult pig. So it stated that it's a lot of similarity in other study in the methodology were used to isolate and culture the pig neural progenitor cells, astrocyte, and microglia. And beside that, it also mentioned about the other study which isolate and culture the endothelial cell from the pig brain microvessel. And what's special about it is it involves the proco density centrifugation for isolation of specific cell. But however, it cannot provide a very high purity. And then another studies that the discussion mentioned about is it involved the culture of the cell before purification. But however, the study is not allowed to analyze a freshly isolated cell, just like the journal provided. And then lastly, it stated that none of the reported methods allowed to purify all different gland and endothelial cell from an adult pig in parallel and give access to freshly isolated as well as culture cell. And last but not least, in summary, the journal stated that it provided inexpensive and comprehensive method for primary gland and endothelial cell culture. And what is the summary? It can be divided into 4, which is the gland and endothelial cell were prepared in bulk, which allowed to set up co-culture experimental and investigate different gland and endothelial cell in parallel. And is also able to isolate and culture different types of gland cell and endothelial cell from a single pig brain in parallel. And then the functional integrity of the cell was proven by different cell types specific assay. And last but not least, cell were maintained in culture for up to 8 weeks without significant change. So this is the summary of the discussion. Yeah, so I guess that is our group Mindmap presentation. Thank you for your watching.